So students, today we are going to study reticulocyte count. Reticulocyte count is one of the blood test, hematological blood test, and reticulocyte. If you see, if you want to know the little bit information about reticulocyte count or reticulocyte, so we can know some background about reticulocyte, as reticulocytes are non-nucleated RBC that still contain RNA. This is the typical characteristics of reticulocytes. We can differentiate the mature red blood cell and immature red blood cells microscopically, and reticulocytes are immature red blood cells and usually contain RNA in their cytoplasm. Okay, so you have to remember that reticulocytes are uh, immature form of the red blood cells, and in their cytoplasm we can see RNA, and after staining we can be able to trace out the RNA material. It's usually you can see in the image also. There are two types of cells in the image. One is the cells. One cells are uh, usually don't have the bluish color, dark bluish color in the cytoplasm. So the cells lacking bluish color in the cytoplasm is usually the mature red cells. But the cells which contain clusters of blue color, dark blue color in their cytoplasms are usually the because of the RNA content, and those cells are reticulocytes. Reticulocytes are usually present in blood for about 2 days before developing into mature red blood cells. This is what it takes 2 days to become red blood cells from reticulocyte. So during the blood formation, basically during the formation of red blood cells, erythrocyte is a second last stage. Okay, last stage is the RBC, which is the mature form of what we get. But before maturation of red blood cells, the pre-stage or pre-maturation stage is the reticulocyte. Okay, reticulocyte count helps to assess therapeutic activity of the bone marrow. The bone marrow is the place where blood cells are produced. The blood cells, basically RBC, WBC, and platelets. All cells, blood cells, are produced in the bone marrow, and the process of formation of red blood cells is called erythropoiesis. So, erythropoietic activity, if you want to assess the function of bone marrow, basically the erythropoietic activity function of the bone marrow, then we can perform reticulocyte count. It gives some idea about that the bone marrow is functioning properly, or maybe there is a compromise in the functioning of the bone marrow. Okay, we can visualize reticulocyte count, or we can visualize reticulocyte by staining with the supravital dyes or supravital stain. Okay, so the stain usually we consider is a new methylene blue or brilliant casyl blue. So these are the available stain, okay, that contain dyes, methylene blue or brilliant casyl blue dyes. So these dyes are basically they stain the RNA material present in the reticulocyte and helps to differentiate reticulocyte from red blood cells. To perform this experiment manually in the laboratory, we require some requirements like syringe, blood vials, microscope, water bath, glass slides, test tubes, master pipettes. And these are the composition of beryllium crescent blue reagent, which is 1% beryllium crescent blue. And there are, apart from beryllium crescent blue, there are other components are there. The composition is we have to take 1 gram of beryllium crescent blue, 0.4 gram of sodium citrate, and 0.85 gram of sodium chloride. So this component, these solutes, you have to dissolve in 100 ml of distilled water. Okay, sodium chloride basically it maintains the isotonicity, means it prevents the blood cells, red blood cell, from destruction or hemolysis. So it maintains normal, isotonicity. And sodium citrate, it prevents the aggregation of red blood cells and also prevents clotting. Okay, and brilliant crescent blue, this is the basic dye which can bind to the acidic part of the cell. So RNA is the acidic part of the cell, so it has an affinity for brilliant crescent blue, and because of that it can bind with the RNA, and RNA material can be seen blue color. What sample do we require for reticulocyte count? So we can take whole blood, that is anticoagulated blood, and we prefer EDTA or heparin as anticoagulant. We can also take capillary blood, and you can put in the heparinized tubes, or immediately we can take the blood from the capillary and we can mix with the stain and we can perform the procedure. Okay, the sample is a whole blood sample. Okay, principle of the method is the reticulocyte count is based on supravital staining, where reticulocytes are visualized by staining with the supravital stains. What is the meaning of supravital stain? Here, the stain is designed for staining living cells. So we should take the fresh sample and we should mix the blood sample with the stain. And in the living condition, the red cells should be, or the reticulocytes should be stained. When you take the blood sample and if you store for longer periods, then it may gives incorrect results. So it is advised to take the fresh sample always. And this stain causes the ribosomal and residual RNA to precipitate. So the RNA present in the cytoplasm of reticulocytes, so they usually bind with the stain, the dye present in the stain, and after that the complex of the RNA with the dye become start to precipitate. So blood smear is made to study red cell and reticulocytes. So when we after mixing with the this supravital stain, what we need to do, we need to prepare blood smear, and after that we can study both red cell and reticulocyte under microscope. Microscopically, dark blue clusters or filament-like structures are visible in retic cells. So this is what we can observe in reticulocytes. Reticulocytes are also called retic cells. So these clusters of dark blue or filament-like structure can be visible inside the retic cells. And this differentiates retic cells from the red cells. Okay? So retic cells and red cells can be differentiated based on this observation that dark blue clusters or filament-like structure can be visible in the cytoplasm of red cells, retic cells. The number of red cells and reticulocytes are counted under oil immersion objective at least in 15 fields. So you have to observe at least 15 fields in the microscope at high power objective, or maybe at oil immersion objective, that is 100x. With help of oil immersion, you have to observe the red cells and reticulocyte, and you have to count it. So the procedure you can do it. You have to take 2 drops of blood in a clean glass tube. This is the first step in the procedure. So only 2 drops of blood is required in a test tube. The fresh blood should be considered. Add equal amount of stain in it. So the stain that you have taken as a requirement. So you can take equal amount of the stain. So blood and the stain should be mixed in the ratio of 1:1. Okay, equal volume. Then keep at 37 degrees Celsius temperature in water bath. This is the incubation temperature where you can incubate it or uh, for at least for 30 minutes. Okay, at least. So during this period, at this particular temperature, the stain enters into the cytoplasm of reticulocytes, and if there is RNA, then it binds with the RNA, and under the microscope we can be able to find it out. Now, after the incubation, take out the tube from the water bath, mix well, and make a smear. So you have to prepare the blood smear. So a smear, there is some special procedure is there to make this smear. There are thin smear, thick smear. So you need to do some practical, some procedure. It's a hands-on training you have to do so that you can become an expert to make a smear. Okay. Once you make a smear, the good smear, then dry the smear now and examine the smear under oil immersion objective. So when you observe under the microscope, so observation is retic cells usually should be stained deep blue. So reticular material, basically we are talking about the reticular material, the RNA material. They should be stained with the deep blue color, the clusters of blue on deep blue color, and it means the cell is retic cells or reticulocytes. Red cells show no bluish coloration. Okay, so this is what observation. Now we can count. We have to do counting now. If you are able to differentiate between retic cell and red cells, now we need to do counting. So choose an area of the field where the cells are exactly seen. So first of all, you have to choose one area, okay, one of the field, and you have to start. You have to check that the cells are visible or not. So using 100x oil immersion lens, count the number of reticulocytes seen per 1000 red cells. So you have to change the field and at the different field, at least 10 to 15 fields you have to observe, and you have to count at least 1000 red cells, and among 1000 red cells how many reticulocytes were seen. So you have to record it. Okay, so using this can be done as at 100 while 100x objective. Okay, at 100 oil immersion of the lens, and once you have done the counting process, then what you do, you do the calculation. So count at least 10 fields. Okay, it is suggested that you need to count at least 10 fields under oil immersion and look for retics against 500 or 1000 RBC. So when you count in one field, first of all, when you count in one field, so you might be able to find red blood cells, numerous red blood cells, and a few reticulocytes. So in every field, you have to count red blood cells and retic cells. So let's say you have counted 500 or 1000 red blood cells in different fields, and also you have recorded the number of retic cells. Now you can use this formula for calculation. Calculate as follows. The retic cell formula is number of retics counted divided by number of RBC counted multiplied by 100%. Okay, so multiply by 100, so the value it comes in the 100 percentage actually. Okay, so you have already counted the number of reticulocytes and number of RBC in 10 different fields. Okay, and after that you can multiply with 100, and the value that we will get, it comes in the percentage. And this is the normal range. The adult usually have a 0.5% to 1.5% retic cells. This is the normal. And newborn baby have retic cells around 3% to 6%. So this is the normal range of retic cells in in their blood. Now, clinical significance. Why do we perform this test? What is the significance? Here we can see that reticulocytosis is the one term you need to know. It means the reticulocyte count rises when there is a lot of blood loss. So during blood loss, usually blood loss occurs during accidents, maybe during the surgery, operation. Okay, uh, so blood can be lost. In a lot of blood can be lost, and another condition is in certain diseases in which red blood cells are destroyed prematurely, such as hemolytic anemia or malaria, where RBC is lysed by the parasites, like incompatible blood transfusion, where blood is not compatible, and uh, then it causes immune reaction, and blood cells can be hemolyzed in the blood vessels. So there are two conditions where reticulocyte count can be raised. The first condition is blood loss, excess blood loss, and the second condition is there are certain diseases where red blood cells are destroyed prematurely. Okay, so the two conditions can can leads to increase the reticulocyte count, and another conditions can where reticulocytosis can occur is high altitudes. When you go to high altitudes, uh, like a mountain region, so at high altitudes, reticulocyte counts may rise. This helps to adjust to the low oxygen level at high altitudes. As when you go to the high altitude, the oxygen levels are low. So to compensate with the low oxygen levels, your bone marrow start to produce more red blood cells. Okay, so at high altitude, reticulocyte count may can increase. Okay, so that can adjust the low oxygen levels. Level less than 0.5% represent inactive bone marrow. When bone marrow is inactive, in such condition, reticulocyte count may be less than 0.5%, and high level of reticulocyte count may represent hypererythropoietic activity in bone marrow. Okay, bone marrow may show hypererythropoietic activity. So in such condition, high level of reticulocyte count may be seen, and this hypererythropoietic activity can occur when there is a excess blood loss or when there is a premature destruction of red blood cells. So usually these two conditions there is a hypererythropoietic activity. So we have studied reticulocyte count, the principal, procedure, methods, requirements, and now we have come to the end of this presentation. Now we can discuss about doubts if we have.